And so what we're going to do today is we're going to pick up, of course, where we left off last time, and we're going to start talking about the structure of phospholipids. And we introduced this last time. We described how the phospholipids are derived from triglycerides, that two of those fatty acid side chains remain the same. But on the third carbon is modified in such a way that rather than linking through an oxygen to a fatty acid, it links through an oxygen to a phosphate. That phosphate then, in turn, is linked to a serine, a threonine, or an ethanolamine, but in all cases, they're charged. So now this derives then a structure which gives us two fatty acids and one phosphate head group, and because of the phosphate head group being charged, this molecule is amphipathic. We'll begin the discussion here today. We can imagine a kind of thought experiment where we take a salt shaker full of these phospholipids and we shake them out on the surface of water and ask simply the question of how do those phospholipids orient themselves on the surface of the water. And I think you'll see simply that the phosphate head groups will face the surface of the water and the tails will turn away. And that works fine. It's a kind of thought experiment in thinking about the orientation of phosphate head groups with respect to water. But it doesn't tell us very much about how these phospholipids are going to function as biological membranes, because, after all, biological membranes live in an aqueous environment. They have to be immersed in water. So we'll work our way through this sort of intuitively, and we'll then talk about first sprinkling the phospholipids on the surface of the water, but then we're going to talk about what happens if we try to dunk those phospholipids beneath the surface of the water. The first low-order state we can think about is something called a micelle. And in the micelle, the tails turn inward and the heads face outward, and you can, for short durations, have these micelles, where you'll have a sphere of phospholipids with their hydrophobic tails pointing inward and their hydrophilic heads pointing out to the water. The problem with these micelles is they tend to be unstable. The problem is the least little water around those hydrophobic tails and the whole thing just blows up. So they're not very long-lived. In order to get a stable state, we have to form some kind of a bilayer. And we'll walk our way through this first by forming a liposome, which is really a sphere. It's a sphere where we have a lipid bilayer so that we can have a well of water on the inside and maintain a stable state. But the liposome is really a small sphere, and what we really have to think about is how we're going to generate cells. So we'll try, by looking a little bit at dimensions, go from a liposome to a planar bilayer. One of the characteristics of all of this will have to do with the small size of these molecules. We're going to be thinking about things on a molecular level. And we have to go from thinking about the structure of molecules to begin thinking about the structure of cells. And we'll find there's about 1,000-fold difference in the size of these individual molecules and the size of a cell. And we'll talk about that a little bit. We'll generate then a phospholipid bilayer, and this is a stable structure, and it forms a formidable barrier between the outside and the inside of a cell. But biology requires communication between the outside and the inside and the outside of a cell. And so we have to then think about a way that we can communicate across the planar bilayer. The communication across the planar bilayer will be by transmembrane proteins. What we're going to see is that biological membranes are organized into something known as the lipid mosaic model. And what we mean by the lipid mosaic model is we have a great planar sea of a bilayer of phospholipids. But that layer, that two layers of phospholipids, is now traversed by a protein. These will be transmembrane proteins. So we'll now think about having the transmembrane proteins communicating from the outside to the inside of the cell. Now we'll lay this out first, showing these transmembrane proteins more or less as potatoes extending across the lipid bilayer, which of course can't be biologically correct. But to think about these in a biologically correct fashion, what we really need to think about is a little bit of protein chemistry, so we'll fall back onto this idea of generating proteins. We'll generate proteins first by talking about the primary structure of a protein, where we're going to take individual amino acids and make polymers of these amino acids to make a protein. and we'll describe the primary structure of a protein as simply the sequence of the amino acids in that protein. By generating and looking at the structure of amino acids and how they're linked one to another by peptide bonds, we'll see that there's a commonality of how we make these polymers. We're going to take one amino acid and always, always, always link it between its amino end and its carboxylic acid end. This linkage will be something known as a peptide bond. What this will do then is it's going to show us that all proteins have in common these amino acids linked by peptide bonds. All amino acids have an amino end and a carboxylic acid end, but they also have a third important characteristic. Every amino acid will have something known as an R group. The R group can be as simple as a single hydrogen. It can be an acetate. It can be a complex ring structure. There are any number. In fact, there are 20 different R groups. The 20 different R groups determine the 20 different amino acids. But we'll see then, though, that the amine and the carboxylic acid structure will be important for forming the peptide bonds, and the R groups will be outliers for the moment. We're just going to put the R groups on. That will then generate the idea of a single string of amino acids, a polymer of amino acids, the primary sequence of amino acids, the primary structure. That's simply like taking amino acids and naming them and putting them out like pop beads on a string. That's the primary structure. But proteins are far more than just a string of amino acids. For proteins to function in biology, they have to have a particular three-dimensional shape. So we'll work our way through how we get this three-dimensional shape. The first level of shape that we'll talk about is something known as the secondary structure. The secondary structure derives entirely from hydrogen bonding within the molecule. The secondary structure derives from the fact that this hydrogen on the amine is positively charged, and the fact that the oxygen on the carboxylic acid, that's so-called carbonyl oxygen, is negatively charged. We can use the negative charge on that oxygen and the positive charge on this hydrogen to now begin to orient this molecule into a particular shape. We're going to find that this hydrogen bonding between the amino hydrogen and the carbonyl oxygen forms the secondary shape, and the secondary shape can take two kinds of structure. It can either be an alpha helix, a kind of spiral staircase, or it can be a beta pleated sheet, that is a zigzag, and we'll talk a little bit about this detail. The alpha helix and the beta pleated sheet provide the first beginnings of the three-dimensional structure of the protein. But in itself, the secondary structure, because it derives only from hydrogen bonding and does not involve the R groups, the secondary structure in itself is not enough to determine the three-dimensional shape of a protein. The three-dimensional shape of the protein largely comes about because of the R groups. Now, there are 20 different R groups. Those 20 different R groups can be hydrophobic. They can be a single hydrogen. They can be an acetate. And they can largely lack charge, so they're uncharged. Other R groups will be polar. And beyond that, some R groups will be truly charged. So now you can begin to see here that we're now going to have R groups, some of which are hydrophilic and some of which are hydrophobic. And now the characteristics, either hydrophilic or hydrophobic, of those R groups are now going to allow those amino acids either to face the water or face inward away from the water. And that's then the beginning of the generation of the three-dimensional shape. We'll try to walk our way through sequentially what's going on here to generate the three-dimensional shape of the protein.